So, it started with not, and what is it next?
Yeah, the next is what I just mentioned, the scid mutation. So the scid mutation, it was first arose in the CB17, which is pretty similar to the BAP-C background.
Yeah.
So it's a spontaneous mutation that was found in the CB17 colony.
Okay.
And at that time, people do not have the genetic engineering tools as much as we have today. So one thing people trying to study the mutation is by putting the strain and putting the mutation onto different strain background. So accidentally, when they put the scid mutation on the NOD background, it turned out it's actually works better than the CB17 in two different ways. Because the CB17 has a thymic lymphoma, but when it was moved to the NOD background, it does not develop thymic lymphoma anymore. And but more importantly, in the CB17 background, the scid mutation has leakage, which means that the B cell and the T cell will develop, even though at a reduced proportion, but they still have the B cell and the T cell towards the later stage. But when it was moved into the NOD background, the leakage is gone, meaning that the penetration is 100%.
So when you say the spontaneous mutation, it's a gene, right?
Right.
What is it again?
The gene itself is called the Prkdc.
Prkdc.
Yeah, it has a long name, but what essentially is that when the B cell and the T cell develop, they need to undergo a process called the VDJ recombination, which to make a functional B cell receptor and a T cell receptor. And scid or Prkdc is one of the enzymes involved in that process, because when it makes a functional B or T cell receptor, it undergoes this VDJ process that first make an double-strand DNA break and then have to, so it cut and then paste. So the Prkdc is one of the enzymes involved in DNA repairing. So without the proper repair of the double-stranded DNA, the B cell and the T cell cannot mature.
So the scid mice, this kind of repairing enzyme is kind of defective.
Yeah.
And then when you breed this with the NOD, you come up with the non-scid.
Right, which by itself is immunodeficient mouse model widely used.
And then